Okay, let's make a start. It's 3:30. So if you could close the door at the back there, please. Okay, so it's been a while since our last lecture, and I will just remind you about our last microbe of the week. And I gave you a tricky question about sulfur disproportionation. So some autolithotrophs can gain energy by sulfur disproportionation. And the question was, what is that? What is the electron acceptor? What is the electron donor? So this is a paper from 1993. When this was published, it was the discovery of a new type of lithotrophy. So the reaction is shown here. Okay, you start with elemental sulfur, so it's S8. Okay, 8 sulfur atoms in a ring structure. That's elemental structure, sulfur. And it is disproportionated into sulfate and hydrogen sulfide. And the question is, what is the electron acceptor and what is the electron donor? So I had a lot of confusion on this point, not surprisingly, because it's tricky. Okay, but you see in this diagram, okay, what is the oxidation state of sulfur? It starts out as 0 in the elemental sulfur. Some of it becomes oxidized, okay, loses electrons and gets an oxidation state of +6. And some of these sulfur atoms are reduced. They end up with an oxidation state of -2. So in this case, the elemental sulfur that you start with is both the electron acceptor and the electron donor. Okay, not the same atom, okay? Some of these sulfur atoms lose electrons, and they are the electron donors, and some of them gain electrons, and they are the electron acceptors. Okay, so that was the tricky part of the question. And some people kind of got it the wrong way round. You said that, okay, the sulfate was the electron acceptor and the hydrogen sulfide was the electron donor. But that can't be right because they are the products of the reaction. They're not the substrates. Okay, electron donor, electron acceptor, they're always going to be a substrate. They're never going to be a product. Okay, now, I don't know about you, but when I see something like this, I kind of think this just doesn't make sense. It should never, ever happen because elemental sulfur is quite stable. Okay, it's not going to spontaneously degrade into these two different parts. And in fact, that's true because in the section in here, they talk about the thermodynamic considerations. You can see that disputation of elemental sulfur to sulfide and sulfate is endergonic. So it's never going to go in the right direction. How can you get energy from an endergonic process? Normally it can't be done. But this is the trick, okay? The delta G is strongly dependent on the concentrations of the sulfide and protons. So if you've got very low concentrations of sulfide, then the delta G is sufficiently low that you can gain energy through the reaction. So it means that this is going to generate energy as long as you can get rid of the hydrogen sulfide. And it works because it will react with iron. So this precipitates out. So the concentration of sulfide in solution is always very, very low. So this can only possibly work as long as you've got enough iron 2 in solution. And it will precipitate out into iron sulfide and pyrites. So that's how it works. And why is this interesting? You might not think it's very interesting, but I do, because in 2011, bacterial microfossils were discovered. They're the oldest microfossils which have a consensus among the specialists that they do represent actual bacterial cells. And next to these bacterial cells, they found crystals of pyrites. So that was an indicator, and the authors of that paper suggested that those bacteria 3.5 billion years ago lived by using this kind of chemolithotrophic metabolism. And it would work because you don't need molecular oxygen at any step. Okay, the atmosphere of the Earth 3.5 billion years ago was highly reducing. Most chemolithotrophy that we know requires molecular oxygen, but not this one. So it could work in the conditions of the early Earth. Okay, so that's why I asked you this question about bacterial metabolism. And the scores on the doors are up here, okay. That's what I have. I'm afraid she has left us. She is no longer with us, Jolan. So, you know, she's knocked out of the race, so that anybody with more than 5 points so far can probably win the Microbe of the Week. So we will do another one this evening, okay? I will send out the question at 7 p.m. And don't forget, there is the bonus for the first correct answer. A lot of people send me answers, but 1 week later, it's correct, but you're not going to get the bonus. Okay? All right, that's it. Okay. Our focus is going to be bacterial pathogens. And for today, we're going to do the first part, definitions, characteristics of bacterial pathogens, physiopathology, how does physiopathology a little bit, but how does a bacterial infection progress, and what does it take, what distinguishes a pathogen from a harmless bacterium? How do bacteria become pathogenic? What are the functions that allow them to infect us and cause disease? That's what I mean by physiopathology here. And hopefully I will have time to also cover host defense against infection at the end of this afternoon. And then on Monday, I'll be illustrating with some examples. Okay, so always a good place to start is Koch's postulates. How do we know when somebody has a disease? How do we know that it's caused by a bacterium or any other type of infectious agent? Also valid for parasitic diseases and viral diseases. So this goes back to the 19th century when Koch and Pasteur were promoting the germ theory of disease, which held that transmissible diseases were caused by infectious agents, bacteria or viruses. And they had a lot of resistance from the medical community at that time. So they had to try and really prove beyond any doubt that when they got a bacterium, it really was the cause of the disease in people. And it was really Robert Koch who put these rules down in a clear way. So he said that you can be certain that a microorganism causes disease if it's found in all patients with the disease, and of course is absent from patients who do not have the disease, and it's found in the affected organ. Okay, so if you've got a lung pathology, bacteria should be in the lungs, not on the skin or something like that. Now, once you've documented these two things, you might think that's it, we've done our work. Okay, it's the bacteria, it's there, all the people who've got the disease, they have the bacteria, nobody who's healthy has it. That's it, it's been proven. But that's not true. If you do that, then you have demonstrated a correlation. You've shown that the presence of bacteria are correlated with the disease. And correlation is not the same thing as causation. And that was exactly the objection of some members of the Academy of Medicine who said to Pasteur, yeah, well, of course these people have got bacteria. They're sick, you idiot. They're more susceptible to bacteria because they are sick. They are not sick because they have the bacteria. The causality is the other way around. Okay? So if you want to prove that the bacteria cause disease and are not just correlated with disease, you have to go a step further. And you do that by performing, just like in any kind of scientific domain, you perform a controlled experiment. Now, in order to do that, first, you have to get a pure culture of the agent. Okay, you have to get a sample from the sick person, spread it out on an agar plate, pick an isolated colony, get a pure culture. And then you re-inoculate it into a healthy animal, or perhaps a volunteer, depends on the severity of the disease. And if it produces the same symptoms, then you can be sure that your bacterium has caused the disease, because you're controlling what has been done. You have a healthy person or a healthy animal initially, you give them the bacteria, and you can show that way that the bacterium is the cause of the disease. Now, just to get the kind of cherry on the top, you can also re-isolate the same organism from the experimentally infected subject, just to really prove that it's no accident. So if you do all of that, then you've proven really without any doubt that your microorganism is the cause of disease. And I like this very much because it's a kind of beautiful miniaturized example of the scientific method. Okay, you start out with a correlation, you establish a correlation, but that's not enough. Afterwards, you have to perform a controlled experiment, and then you really prove something. So those were the principles set out by Robert Koch, and they still are the gold standard for determining whether a microorganism causes disease. However, it's not always possible to do or to validate each of these 4 points for every microorganism. So, for example, sometimes some bacteria can secrete exotoxins that can cause pathology at a site distant from where the bacteria are. So in that case, you won't be able to prove point number 2 here. Sometimes you have healthy carriers. This was not known at the time of Robert Koch's work, but subsequently for many diseases you can have people walking around, they're infected with a bacterium or with a virus, they can shed it, be infectious to other people, but they themselves are healthy. So, okay, point number 1 has got to be modified slightly. So it doesn't have to be 100%, 0%, but you have to have a higher incidence of the presence of the microorganism in the people who are sick. And often, for point number 3, it's impossible to do. Okay, for this one, you have to be able to obtain your A pathogen in pure culture. For some viruses, it was very, very difficult. Hepatitis B virus was very difficult to cultivate. Human papilloma viruses, also very difficult to grow in culture. And then, of course, if you don't have an animal model, then it's not ethical to infect people experimentally with dangerous bacteria or viruses. So it might be practically or ethically impossible to validate point number 3. Okay. So this is the, you know, in the best case scenario, you can validate all of these points, but in many cases, one or more of these points are very difficult to establish. Okay, so that's how you know that a bacterium causes disease or not. And once this has been done, you can, one finds that the types of bacteria that cause diseases are either strict pathogens or opportunistic pathogens. Now, strict pathogens only cause disease. You can only find them in the context of people who are getting sick. Whereas opportunistic pathogens, most often they don't cause disease. They have another mode of life and from time to time can cause disease. Okay, so for example, Mycobacterium tuberculosis is a strict pathogen. It can only grow in humans. And in people, it is always associated with pathology. An opportunistic pathogen, for example, Pseudomonas aeruginosa. Opportunistic pathogens, there are 2 types. They are either pathogenic strains of commensal bacteria, for example, Escherichia coli, Staphylococcus aureus. You know, we have a lot. Everybody here has got Escherichia coli in their gut. Most strains are commensal and harmless. Some are pathogens, and some other strains are pathogens. And from time to time, saprophytes, who will live by degrading organic matter, can be opportunistic pathogens. So Pseudomonas aeruginosa is one of those. So particularly with opportunistic pathogens, different strains, different isolates will have differing virulence. What does that mean? That means the ability of the organism to cause disease. And there's kind of 2 main components of this. There's the ability to infect and invade new hosts and the capacity to cause damage to the host. So a highly pathogenic bacterium has got to have both, okay? Something that is highly infectious but not very, very dangerous. They can be very efficient for infecting people, but perhaps not really too nasty once you do get infected. And experimentally, you can measure these 2 aspects by the ID50, the 50% infectious dose, or the LD50, the 50% lethal dose. And of course, different strains of the same species of bacterium can be more or less virulent. Okay, so you can have many strains of Staphylococcus aureus hanging around in your nasal cavity. They will not be very virulent. Some in perhaps causing infections in a hospital ward, they might be more virulent. Okay, so experimentally, how do you measure the 50% lethal dose? You get a bunch of mice and you say, I'm going to test different doses and I'll inject 10 mice at each dose. And then over time, I will count the number of animals that die or that display symptoms. So if you're counting deaths, then you will determine the LD50. If you're counting the apparition of symptoms, then you'll determine the 50% infectious dose. So you'll generate a curve like this. So this is the rate of mortality or morbidity if you're just looking at symptoms, and is a function of the injected dose. So at a very low dose, none of the animals will die or get infected. At a very high dose, all of them will be infected. And the 50%, the LD50, is where this curve cuts the 50% line here. So for different bacteria, you have these kind of numbers. And you can see that an ID50 can go from, I don't know, less than 100 CFU, colony forming units, up to maybe 10 to the 10. So you have 8 logs difference here. Okay, and of course, the lower this value is, the more infectious the bacteria are. So when this is low here, that means you are what is this in English? Anybody? Yeah, what's that in English? Guinea pig. Okay, if a guinea pig inhales 100 bacteria here, it will get infected. So the bacteria are highly infectious. If the ID50 is very, very high, the bacteria are not very infectious. Everybody understand this? Okay. Okay. So that's how you measure how pathogenic, how virulent, and how infectious different bacteria are. An ID50 and an LD50 test. Okay, so now in terms of physiopathology, what does a bacterium need to do to infect a host and cause disease? Well, the first thing, got to be transmitted. Once it arrives at the new host, it's got to be able to colonize that host. Once it's, managed to attach to the host tissues, it's got to be able to grow on or in the host. Of course, we are not going to sit around waiting to be eaten by bacteria. So any type of host, animal or plant, has defenses against microorganisms. So a successful pathogen will be able to escape from the host defenses. And finally, to really cause disease, to cause damage, there's got to be some specific mechanism by which the bacterium actually harms the host. So these 1, 2, 3, these points 1, 2, and 3, they're to do with infectivity. Okay, the ability to be transmitted and colonize a new host. And 3, 4, and 5, these are to do with pathogenic potential, the ability to cause disease and harm the host. Okay, so there's a little bit of overlap between these two, because growing inside the host is always going to be a little bit harmful. Okay, it's going to have some negative consequences. If we're talking about, a body compartment that's normally sterile, it will always be harmful. Okay, so first thing, transmission. Get transmitted to a new host. Okay, one of the things you can think about is whether transmission is direct or indirect. Direct transmission is, I've got a bad cough, I'll suddenly explode in a fit of coughing, and everyone on the front row gets infected. Direct transmission from one host to the next one. Okay. Carinobacterium diphtheriae, Mycobacterium tuberculosis have direct transmission. Indirect transmission, one infected host contaminates something else, and the next host comes into contact, not with me, for example, but with whatever material I've contaminated. So it could be water, Vibrio cholerae contaminates water. It could be the ground, okay? Bacillus anthracis, spores persist in the ground. It can be food, food-borne diseases, Listeria monocytogenes. So this will be contaminated food, will be the link in the transmission chain. Okay, routes of entry. Well, mucosa are very susceptible to infection, so your oral mucosa can be the site of infection. Okay, food-borne infections, Listeria, Salmonella, and Vibrio cholera from drinking water. Okay, respiratory mucosa, our route of infection. Cutaneous infection, not so frequent, does exist sometimes. So your skin's actually quite a good barrier against most bacteria if you don't have a cut, okay, if your skin is intact. Very few bacteria can actually come in and harm you. There are some exceptions. One of those is Rickettsia typhi, Rickettsia provozekii, which cause typhus, okay. The clue is in the name. So these are spread by infected lice. Everybody know what lice is in French? Anybody know how typhus is spread? You had a lot of it in the First World War in the front line. Yeah, it was a terrible cause of death in concentration camps in the Second World War. Yeah, you're on the right track, but it's not punaise. Rats have it, but they're transmitted to humans by? No. Well, okay, yeah, I think cats can also be a host, but to get from the, not exactly puce, but something like that. Yes, the poux. Lice, yeah. Clothes lice, because there are 3 species of lice that can infect humans, right? Head lice, genital lice, and clothes lice. These infections are transmitted by clothes lice, okay? So people living in unsanitary conditions can have lice in their clothes. Okay, so that's what it is, okay? The bacteria grows inside the louse in its gut. And when it bites you, after it's bitten you and had a blood meal, it will crap on you. It's going to defecate on your skin just after it's bitten you. And that's full of Rickettsia bacteria. So afterwards, you scratch the bite, and that's how you get infected, because you rub the bacteria into your skin. Yeah, so if you can avoid scratching, then you have less chance of getting infected. Okay, so that's cutaneous infection, rather rare, okay? Most infections that start in the skin have to have a kind of puncture of the skin at some point. So this is transcutaneous. Either you have a cut, or you prick your finger with something like a thorn, okay? And of course, you know, tetanus is the big one, Clostridium tetani. You know, working in the garden with old rusty tools or something, you can get tetanus. If you get a cut and it's infected, you know, it can be streptococcus, can easily give you an infection. But the skin barrier has got to be damaged for this to occur. And last but not least, sexual transmission. So the genital mucosa are often sites for infection. Okay, Treponema pallidum causes syphilis. Neisseria gonorrhea causes, what is okay, gonorrhea, I guess, sometimes can be asymptomatic or mild in men. You can have a very severe burning sensation when you pass urine if you have a gonorrheal infection. But sometimes it can pass, you can still be infectious. For women, you can have gonorrhea. This can develop into salpingitis and scarring of the fallopian tubes. So it can be a cause of pelvic inflammatory disease and can cause sterility. Often because the male partner did not have a symptomatic infection, didn't know he was infected, transmitted it to his partner. Okay. Okay, quick question while we're on this subject. Who says true? Who says false? Yeah, you're right, it's false. So Bacillus anthracis is a kind of example because it's a strict pathogen, but it can persist as an infectious spore. So that's what allows indirect transmission. Okay, so these are transmission to a new host. Yeah, one more thing about that, okay? Okay, why do bacteria make you ill? Well, it's to do with transmission again, okay? Because, you know, these bacteria that want to infect people via the respiratory tract, they make their hosts cough, okay? Once you're infected, you cough. And this is going to favor transmission to the next person, okay? Vibrio cholerae wants to be transmitted, so they cause diarrhea. It will favor transmission to a wide number of new hosts. So sometimes, you know, symptoms are directly related to the transmission cycle. Okay, right. So somehow the bacteria are going to arrive on a new host, the gastrointestinal tract or the respiratory mucosa. What they've got to do is they've got to adhere to the host cells. And of course, this is not an accident. It's mediated by bacterial adhesins that are either fimbriae, okay, filamentous structures on the outside of the cell, or non-fimbrial adhesins. Okay, so just a reminder what fimbriae are. Filamentous structures often involved in adhesion to biofilms and inert surfaces. Now, this can play a very important role in the tropism of the bacterial infection. That is, where the bacterium can cause infection. So, in Escherichia coli, there are uropathogenic E. coli and enteropathogenic E. coli. And uropathogenic E. coli express a fimbrial adhesin that allows it to adhere to epithelial cells in the urinary tract. So that's why this strain can cause urinary infections, whereas enteropathogenic E. coli has adhesins that allow it to attach to epithelial cells in the gut. Same thing for Neisseria gonorrhea. It's the adhesins that allow it to start colonizing the urethra and the genital mucosa. Non-fimbrial adhesins, well, this is just a kind of like a catch-all term for any kind of adhesin that is not filamentous. So some pathogenic strains of E. coli use non-fimbrial adhesins. Another one is Yersinia enterocolitica. The non-fimbrial adhesin here is this YAD-A protein. And you can see that it's a protein that is on the outside, covers the external face of the bacterium. So this is YAD-A plus bacteria. If you inactivate this gene, okay, you see this adhesin disappears. So the bacteria have arrived. They've bound somehow onto the host cell. Now they've got to be able to grow in the host. Now, for most nutritional requirements, human body is like the bacterium has died and gone to heaven. You know, everything is there in large amounts. You've got loads of organic carbon, loads of amino acids, peptones, except one thing is not very freely available, and that's iron. Okay, because most of the iron in a human being is not floating around free in solution. It is bound by iron transport proteins. So all pathogenic bacteria must have a mechanism to get iron from the host. And there are different mechanisms that are used. One is to secrete very high affinity siderophores, so they can pick up the very small amounts of free iron that are present. And okay, so Mycobacterium tuberculosis does this, Yersinia does this. Another way of thinking about this for the bacterium is forget about the free iron. We've got tons of iron floating around on these iron transport proteins. So if I just bind one of them and import it, then I can get the iron from the host easily. So pathogenic strains of Neisseria do this. They'll bind transferrin, lactoferrin, and then internalize that and steal the iron. Where else do you have a lot of iron circulating around in the body? In blood. Yeah, red blood cells. So you have a huge amount of iron in red blood cells attached to hemoglobin. So there are pathogenic bacteria that can use the heme group, pick up the heme group and extract the iron. And of course, you have to start lysing the blood cells first to get the iron in that case. So many bacteria are hemolytic. Pathogenic bacteria can be hemolytic, and then they use the hemoglobin that's released to pick up iron. Okay, escape from the host defenses. So the bacteria have arrived, they've bound specifically to some host tissues, they start to grow. So they're producing a kind of little colony somewhere in your gut or in your nose or something like that. Your immune system is going to wake up and realize that they are there and try and destroy the bacteria and get rid of them. So in order to really persist as an infection, bacteria have got to avoid the host immune response. Now, the main thing that's going to destroy bacterial cells are phagocytes, like neutrophils. As we'll see, I hope we'll have time at the end of the lecture. Neutrophils internalize bacteria and kill them. So one of the main strategies for avoiding the host defense for bacteria is to avoid phagocytosis. Prevent the host cells from internalizing them. Now, very, very important virulence factor for many bacteria is the presence of capsule antigens. Okay, so the capsule antigen is going to go around the bacterial cell and hide it from the receptors on the phagocyte that allow the phagocyte to internalize the bacterium. Another strategy is to survive inside the phagocytes. So some bacteria do that. Listeria, Rickettsia, they live inside cells, intracellular pathogens. Okay, so during phagocytosis, you've got the host cell here. There's a nucleus, phagolysosome. Bacteria will be taken up. These two strategies, some manage to survive inside the phagolysosome, others will escape and live in the cytosol. So Salmonella and Mycobacterium tuberculosis can survive inside the phagolysosome. So dodge the host immune response, and finally, to be really evil, you must harm the host. So there are different aspects to this. One of them is to produce hydrolytic enzymes, collagenase, hyaluronidase, and elastase. So this is part of the colonization process. Okay, so imagine this is an epithelium here, apical pole, basal pole. Bacteria will arrive here, start killing these epithelial cells. So they start to grow down here. The next step in the invasion process is to degrade the basal membrane here. Okay, so this is collagen, and hyaluronan. So all of the extracellular matrix. It's got to be degraded to allow the bacteria to start growing. And of course, if they do this, they directly damage the host tissue. Now, there are other enzymes that are produced that have other effects on pathology. So Staphylococcus aureus, it produces a coagulase, so it provokes blood coagulation. So the bacterium gets surrounded by a blood clot. And this is the first step in the formation of what's the word here? Okay, I'm looking for a word. It's escaped me for a second. It will come back in a minute. An abscess. That's it. Okay. Staphylococcus aureus. It's a very specific pathology. Is you get bacteria in the blood and they form an abscess somewhere in the body. Okay, so it's a big kind of pocket full of bacteria that are growing, an abscess. And the beginning of that process is forming the blood clot, and this starts the formation of the abscess. DNase, some pathogenic bacteria produce DNases. Why do they do that? Well, it goes back to this process here, okay? So epithelial cells in a live cell, all the DNA is compacted in the nucleus. If they start to die, then all this DNA here is going to spill out, and it will form a very big, viscous kind of blob. High molecular weight DNA is very viscous. So these bacteria tend to get, caught up in the high molecular weight DNA. To escape, it's part of the invasion process, they produce DNases. Okay, hydrolytic enzymes, one type of mechanism by which bacteria harm the host. Next one is to produce toxins. And there are two types of toxins. Often comes up in exam questions, so you really must memorize this. Exotoxins and endotoxins, what are they? What are the differences? So exotoxins are secreted proteins. There are different families of exotoxins. One of the frequent ones are the AB toxins. They have two components. B is for binding to the host cell, and A is for enzymatic activity. So the big example, the classical example is cholera toxin. So it's a pentamer of the binding subunit, binds onto gangliosides. This will allow the internalization of the bacterial toxin, gets into the cytosol, and then the active part, the A subunit, will cause cyclic AMP to accumulate, and the net result is for this CFTR chloride pump to become activated, and chloride ions will be exported into the lumen of the epithelium. So to maintain osmotic balance, sodium and water follow the chloride ions, and this is directly responsible for the very profuse liquid diarrhea that is the symptom of cholera infection. So it's the toxin that is directly responsible for the symptoms. And if you do not have access to enough clean drinking water, you can die of dehydration, all because of the cholera toxin. So different types of exotoxins have different effects, very specific and often related to the type of pathology that the bacteria cause. Now, endotoxins are different. They are not secreted. They are endogenous to the bacterial cell, and it's because endotoxin is lipopolysaccharide. So all bacterial endotoxins will have similar effects, and they cause fever. Okay, so they're pyrogenic. They cause inflammation. They cause fever. And the reason endotoxins make people sick is because they are recognized by components of the innate immune system. So this is an important part of your immune defense against gram-negative bacteria. It's an adaptive part of the immune system. Oh no, that's the wrong. It's part of the innate immune system, but it's protective if the effect is localized. Okay, so if you have a localized infection somewhere on your finger, then you'll have inflammation. The blood pressure will go down. The endothelial barrier will become more permeable. So this helps cells of your immune system infiltrate the site where the bacteria are and get rid of them. Coagulation will become favored, so you can have a clot that forms and prevent spread of the bacteria. So these are good things to happen if they are localized. But if they are systemic and you have hyperstimulation of the same responses, they can be very, very dangerous. This is what gives you septic shock, because you'll have lower blood pressure, dramatically lower in your whole body. You'll have a more permeable endothelial membrane, an endothelial barrier all through your whole body. And this means that your circulation is going to have a big problem. You'll also have clots forming everywhere. So all the coagulation factors in the blood will be used up, and then this will have the kind of paradoxical effect that you can have hemorrhages all over the place because you don't have any more coagulation factors. And all of this means that a lot of your organs cannot be perfused correctly, so you'll have multiple organ failure as a result of septic shock. So if you have a big infection from gram-negative bacteria that's systemic, you can die of septic shock. Okay, so these are a lot of different processes, okay? Colonization, invasion, avoid the host immune response, and actively harm the host, okay? So for a bacterial pathogen, they really need to have a lot of different proteins that are expressed. Each one of these systems is going to contribute something to pathogenicity. Okay? So if you've got adhesins and a nice way to get iron, then perhaps you can colonize a host, but you won't be very harmful. To be really pathogenic, you also have to secrete toxins and hydrolytic enzymes. So that means that the difference between a harmless commensal bacterium and a pathogen isn't down to one or two genes. It's down to a lot of different genes that are accumulated by bacteria, by pathogenic bacteria. And these genes are carried either on virulence plasmids that can be easily exchanged between bacteria, or they are coded in specific regions of the bacterial chromosome that are called pathogenicity islands. Now, pathogenicity islands, what do they look like? What are their characteristics? Well, often the GC content is lower than in the rest of the bacterial chromosome. They have direct repeats at each extremity. Often they are located next to genes for transfer RNAs. Some of them may carry genes coding for an integrase molecule. And they will have not just one virulence gene, but a virulence operon, okay? Several genes that will be co-regulated. And so the different virulence genes here on one pathogenicity island will code for all the proteins that are needed for one particular system. So an example is in Salmonella, okay? So SPI1, Salmonella pathogenicity island 1, it codes for all the genes necessary to invade non-phagocytes. SPI2 codes for all the genes necessary to survive inside phagocytes. And the major pathogenicity island, SPI7, codes for all the enzymes required to produce the capsule. Okay, so these kind of strange characteristics of pathogenicity islands are characteristic of mobile DNA elements. Okay, so these are transposons or bacteriophage genomes that move about. And that's how they can be transferred from one bacterium to another. Okay. That's about it for the summary. So I'll give you a little question here. See if everybody has understood. So think about these ones. Oh, what's happened? No, come back. Everybody ready? Well, let's see. Okay, for first one, capsule antigens are important virulence factors because they mediate adhesion to host cells. Who says yes? Who says, yeah, one person. Who says no? The answer is no. Okay, the capsule antigen is important to avoid phagocytosis. It's not really an adhesin. Okay, the adhesins are fimbriae or non-fimbrial adhesins, but they're proteins, the adhesins. B, all pathogenic bacteria must secrete high-affinity siderophores in order to obtain iron from the host. Who says yes? Who says no? You're right. There are other mechanisms to do that. C, bacteria with high pathogenic potential have low LD50 values. Who says yes? Who says no? You're right. If you've got a low LD50 value, that means you don't need very many bacteria in order to kill you. So that means they're highly pathogenic if they have a low LD50. So related to that, question D, if a pathogen loses the virulence factor coding for an exotoxin, its LD50 will increase. Who says yes? Who says no? The answer is yes, because it's losing one of the virulence factors that makes it pathogenic, that makes it dangerous, that harms the host. So if it doesn't have this exotoxin anymore, it will be less deadly. So you'll need more bacteria to kill you. You'll be less pathogenic, so the LD50 will be higher. And E, both gram-positive and gram-negative pathogens can secrete exotoxins. Who says yes? Who says no? Who says there's no possible way we can know because I didn't say anything about that? Yeah, okay, yeah, that's true. Most people, yes. So, okay, it's there to remind me because I forgot to say that. So, exotoxins, endotoxin. Endotoxin is LPS, right? So only gram-negatives have endotoxins. Exotoxins are secreted proteins. Both gram-positives and gram-negatives can secrete exotoxins. So it's an important difference between exo and endotoxins. Okay, we have 20 minutes left, so I think we have time to go through host defenses a little bit. Okay, so 20 minutes on host defense, and that will be the end of our work today. Okay, first thing you need to think about are physicochemical barriers that are just physically going to prevent bacterial colonization. So, okay, the skin, as I said, is quite an effective barrier against bacterial infections if it is not damaged. Mucus, it covers the epithelial cells and prevents bacteria from attaching to them directly to the epithelium. And of course, mucus will flow over the epithelium. So in your respiratory epithelium, the cilia all beat in the same direction. They cause the mucus to flow out and expel particles that have landed on the epithelium. So it's a physical barrier to colonization. Peristaltism, okay, same kind of thing in the gut. So you don't have the contents of your intestine just sitting around waiting for the bacteria to colonize you. It's getting pushed through all the time. There's flow, which is important. Also, another thing that's, liquid flow in urine. This is a physical chemical barrier against colonization of your urinary tract by bacteria. Okay, so the pH of urine is not really very helpful for most bacterial infections, and it's always washing things out. Very important, there are 7 times more urinary infections in women than in men, just because the urethra is longer in men. Okay. Oh no, 14 times more frequent. I misread it. So I knew there was a 7 in there somewhere. So just because of this like physical chemical barrier of urine flow, it's a big factor in whether you get urinary infections or not. Another kind of thing like this is for the respiratory mucosa is the way the upper respiratory tract is organized. You don't have a smooth tube that goes from your nose down into the lungs. Okay, I don't really know anatomy, but you know, it's complicated in there, all right? The upper respiratory tract is a complicated structure and it produces turbulent flow. So what that means is that, okay, imagine this is your nose here. And it's going into your trachea. You don't have a smooth tube here, which would cause laminar flow and allow particles to go deep into your trachea and to your lungs. What you have is a kind of complicated system like this, where air that is inhaled is going to end up with a turbulent airflow through the upper respiratory tract. And the result of that is the particles that you inhale will be deposited mostly in the upper respiratory tract, okay, not down in the lungs. And then, because of the mucus flow, they're going to be transported out of the upper respiratory tract into your mouth, swallowed and destroyed. So this anatomic organization is a very important part of why your lungs don't generally get infected. Think about it. In the number of times in a year you get a cold, might be 2 or 3 times a year, might be more. How many times do you get pneumonia in a year? Very, very few. Okay? So it's just because of this kind of physical chemical barrier. Right. That's one thing that's helping to protect us from bacterial infection. The next one is, in fact, the endogenous bacterial flora. So there are sterile compartments in your body, like your lungs, but most of the available surfaces are not sterile. So you've got bacteria all over your skin, as you saw in the lab practicals. Our mouths are full of bacteria. Our guts are full of bacteria. So they're harmless. They're commensal bacteria. But they also protect us from being colonized by pathogens. Now, there are kind of 2 aspects to this, maybe 3 actually. First one is they just take up the space. They are occupying the ecological niche inside your guts. It's already, you know, it's full, guys. There's no space for anyone else to come and live there. Second one is they're actively trying to kill each other. Bacteria in your gut can secrete proteins and peptides that are toxic for other bacteria, called bacteriocins. So Escherichia coli in your gut secretes colicins that are toxic for other types of bacteria. So they actively prevent colonization by other bacteria, by pathogens. And the third thing is they are very important for the correct functioning of the mucosal immune system. So, you know, if you can respond to pathogens when they arrive, it's because your immune system has been primed by commensal bacteria. So the immune system, okay, that's the one that comes last, okay? So probably most pathogenic bacteria that arrive on us, they'll be blocked either by these physical chemical barriers, they'll be blocked by the endogenous bacterial flora, and, you know, it's just the fewer than 1% that get through that will have to be taken care of by the immune system. Okay, so the endogenous bacterial flora, very important. If you have treatment by antibiotics, you can get rid of or seriously damage your gut bacteria. And this can make you more susceptible to colonization by other bacteria. And this is a real problem in the hospital context, where patients who have been treated by antibiotics, perhaps as to prevent infection after an operation, they can become easily infected by other pathogens. So some people have had the great idea to think, okay, if the problem is that they have a defective gut bacteria, and that's why they get infected, maybe we can put normal gut bacteria back into them, and it can be a treatment. So this is the idea behind what's called the fecal transplant. Okay, so what you do is you've got a healthy person here. You get their fecal material, put it in the blender, and mix it up with physiological saline. And then you're going to use this biological resource to recolonize the gut of somebody who's got persistent clostridial infection. So either you put a tube in through the nose, and you can colonize the small intestine and the lower intestine, or you come in through from the other direction and seed the colon. This is a kind of DIY method. I would not recommend this. You're not going to get any further than the rectum here. It will not be efficient. It really needs to be done medically. Now, you might think this is a bit of a joke, but in fact it's not. So this was a publication from the New England Journal of Medicine, published in 2013. It was a clinical trial to treat patients who had a recurrent Clostridium difficile infection. This is the standard treatment, vancomycin. Okay, so they managed to get a 30% cure rate with vancomycin, antibiotic active against gram-positives. This is with fecal transplant, just with one go, 80% cure rate, with a repeated treatment. Almost 95% of the patients were cured of their Clostridium difficile infection. So that's the power of commensal bacteria. Okay. 10 minutes for the immune system. Okay. So in the immune defenses against bacteria, you have innate immunity. And in the innate system, you have humoral and cellular branches. So humoral, so things that are soluble and act against bacteria. Lysozyme, okay, this enzyme which will degrade peptidoglycan. This beta-1,4 bond between N-acetylglucosamine and N-acetylmuramic acid, only present in bacteria. It is not present in any eukaryotic cells. So if you can break this down, you specifically degrade bacteria. Right, now is a good time to remember the cell wall structure of gram-positives, gram-negatives. Okay, lysozyme on its own can only be active against gram-positives, right? Because it will not have can have access to the peptidoglycan on the gram-positive cell wall, but it will not be able to diffuse across the outer membrane of gram-negatives. It's a whole protein. It's too big to go through the porins. So to complement lysozyme, at our mucosal surfaces, different cells can secrete antimicrobial peptides. Two families, cathelicidins and defensins. Okay, defensins are in the gut, okay, intestinal epithelium. You defend your gut. And cathelicidins are in the lungs. Now, the actions of these peptides are to degrade bacterial membranes. So they can degrade the outer membrane of gram-negatives, and this will allow lysozyme to get access to the peptidoglycan layer. Okay, so these two systems work together. And lastly, an extremely important part of our innate defense systems against bacteria is the complement system. Okay, have you seen this in immunology? Yes? They destabilize membranes. I don't know if it will really be important against gram-positives, but I would say that in order to allow other host enzymes to gain access to the gram-negative cells, they are important. So for the purposes of this class, think about defensins with and cathelicidins with relation to gram-negatives. Yes. Okay, complement. Do you know anything about complement? Okay. Complement is a series of proteins in your blood. They're soluble. And they all work together like the kind of blood clotting cascade. Okay, so there are a bunch of proteases. One of them activates the other one, and the end result is you get activation, you get formation of this thing called the C3 convertase. C3 convertase will convert the C3 subunit of complement into C3A and C3B. C3A, you don't need to remember how this works, okay? You just have to remember what happens at the end and what starts it all, okay? The whole cascade is not really our business in microbiology. But what is the end result? Okay, C3A and C5A, it's another part of the complement cascade. These are soluble proteins, and they will attract neutrophils to the site of infection. Okay, so these ones are going to engage the cellular arm of the innate defenses against bacteria. C3b is going to be deposited on the surface of the bacterium. It will covalently react and be covalently bound to the surface of the bacterium. And it will recruit the other complement components that make up the membrane attack complex. So the membrane attack complex will also punch holes in the bacterial membrane. And once again, that will allow lysozyme to penetrate into the periplasm of gram-negatives. Okay, so two effects of complement. Okay, two effects. One of them, attract neutrophils. The second one, get deposited on the surface of the cell and cause the formation of the membrane attack complex. Okay, 5 minutes. I'll just finish complement and then we'll stop there, okay? So how does this whole thing get started? Okay, there are 3 pathways to activate complement. The classical pathway. This is dependent on antibodies. Okay, if you have specific antibodies against the bacterium, they can fix onto the surface of the bacterium, and this will start the complement cascade. The MBL pathway. MBL is for Mannan binding lectin. So the clue here is the word lectin. A lectin is a protein that recognizes specific carbohydrate structures. So these are soluble proteins in the blood, and they can specifically fix to the types of carbohydrates that you find on the outside of the bacterial cell. Okay, that have Mannan. So this is an innate receptor specific for some structures often found on bacteria. And if these bind, that will kick off the complement. And the alternative pathway, this is a really clever one. Right, how does this work, the alternative pathway? I'll try and explain this. Well, okay, the complement cascade is not switched on and switched off entirely, okay? In your blood, you always have a very low level of complement activation, okay? So it's turning over, you know, it's like when you turn your car engine on, it's just going like that, okay? And whether you get complement deposed or not, you have to press on the accelerator. But your engine's already turning over. Now, you might think, why doesn't this explode all of our own cells? Why don't we get complement all over the place if it's already becoming activated? And that's because there are proteins in your blood which are negative regulators of the complement system. Specifically, protein H. So this is a cell in your body. All right, this is the membrane of a cell in your body over here. And we have a lot of glycosylation, and specifically sialic acid, which bacteria do not have. Okay. Neither do insects. I think only vertebrates have sialic acid. So all of your cells that have sialic acid, they will bind protein H. And this is going to stop the complement system. But the arrow is the other way around. We're going to inhibit the deposition of complement on your own cell, and it won't be killed. So think about bacterial cell now. Like this, might be gram negative. Doesn't have the structure that's recognized by protein H. So you'll have a very, very small amount of complement deposited, and then it's a kind of autocatalytic loop, and it will kind of get deposited anyway. So this is kind of recognition of something that is not myself. Okay? The H protein binds on the surface of your cell, says, don't kill me, to the complement system. If this is not there, complement will be deposited. Okay, time's up, so I will send out a microbe of the week tonight at 7 p.m.